Hey everyone, and welcome to the ultimate high-yield crash course on research methodology and medical statistics. Look, if you're gearing up for your final exams, you are in exactly the right spot. Today, we're going to completely demystify chapters 3 and 4 of your textbook. We are taking what can honestly sometimes feel like just a dense, impenetrable wall of definitions, and we're turning it into a crystal clear, logical roadmap. By the time we finish this explainer, you're going to have a rock-solid grasp on exactly how scientific studies are built from the ground up, plus the critical ethical boundaries that keep all that science honest and safe. Okay, let's just dive right into it.
So, here's our syllabus for the session. We'll be moving through 6 distinct phases: gathering the data, defining the goal, picking the lens, designing the study, research ethics, and finally, ethics committees.
Let's kick things off with phase 1, gathering the data. Specifically, we're talking about primary and secondary research. Okay, so every single research project has to start with one big fundamental choice. Where is your data actually coming from? Are you going to roll up your sleeves and dig it up yourself? Or are you going to synthesize what somebody else has already found? Primary research is all about collecting new data firsthand. The whole purpose here is to gather original data that's perfectly specific to your research question. How do you do it? Well, you use methods like experiments, surveys, and clinical trials. Now, because you're literally starting from scratch, it's super time-consuming and costs a lot of money. Think about a researcher running a brand new clinical trial to test a drug or interviewing patients directly about their experiences. That's primary. On the flip side, we have secondary research. This involves taking existing data collected by others and analyzing it. The purpose isn't to gather new data, but to synthesize past findings, maybe spot some trends, or find gaps in the literature. You'll be using methods like literature reviews, meta-analyses, and existing reports. The absolute best part? It's low cost and saves a ton of time. The catch, of course, is that you're entirely limited by the quality of whatever data is already out there. A great example would be reviewing a bunch of published articles on patient care to write a new paper. Moving right along to phase 2, defining the goal. This covers basic, applied, and translational research. So, you figured out where your data is coming from. Now you gotta ask, why are we even doing this study in the first place? Are we just trying to scratch a scientific itch, or are we actively trying to cure a disease right now? Let's walk through the scientific pipeline. Step 1 is basic research. Some people call it fundamental or pure research. This happens in the lab. The goal is strictly to expand general knowledge and figure out fundamental principles. There's no immediate practical application. It's like studying exactly how a specific gene influences cell growth. Then we move to step 2, translational research. This is the crucial bridge. It's all about bench to bedside. You take those incredible lab discoveries and figure out how to move them into clinical use. For example, taking that basic discovery about a cancer gene and transforming it into a targeted therapy. And finally, step 3 is applied research. This is real-world problem solving. It takes those findings and actively uses them to develop new technologies, treatments, or policies. Think about developing a brand new, ready-to-use vaccine based on earlier basic discoveries in immunology. If you're prepping for an exam right now, I highly recommend you mentally screenshot this matrix. It perfectly breaks down the differences. Notice how the settings shift across the 3 types. Basic research happens over in university labs, applied happens out in industries and hospitals, and translational sits right in the middle at medical centers and biotech firms. Also, definitely pay attention to the impact time. Basic research is a long-term, indirect investment. Applied is much shorter term and direct, and translational is specifically engineered for the fast application of discoveries to real-world use. Ultimately, they all form this continuous, incredible cycle of scientific innovation. All right, phase 3, picking the lens. We're talking qualitative, quantitative, and mixed methods. We've nailed down our goal and our data source, but how are we actually going to measure our subjects? Are we hunting for stories and deep meanings, or are we looking for cold, hard numbers? Let's break down your 3 methodological lenses. First up, qualitative research. This is entirely non-numerical. We're dealing with words and themes. The purpose is to really explore human behavior, emotions, ideas, and experiences. You collect this data using open-ended methods like in-depth interviews or focus groups, and then you analyze it thematically, like sitting down and interviewing patients about their personal experiences with alternative medicine. Second, quantitative research. This one is all about the numbers. The purpose is to measure variables, test hypotheses, and find concrete patterns using statistics. You want conclusions you can generalize to a huge population. So data collection is super structured, surveys, experiments, and the analysis is mathematical.
Picture a massive clinical trial measuring the exact drop in blood pressure levels across 500 patients. And third, we have mixed methods research. This just combines both approaches. It gives you a really comprehensive, holistic understanding by integrating numerical stats with deep thematic insights. Think about giving patients a numbered survey to fill out, but then also sitting down with a few of them for personal interviews. You get the whole picture.
Next up, Phase 4, designing the study.
This is where we lay down the actual structural blueprints of our research, and it really boils down to a massive fork in the road when it comes to control. Basically, are we observing or are we intervening? In observational research, the researcher is completely hands-off. You observe the subjects without interfering whatsoever. There are two big types you gotta know for your exams. First, a cohort study, which follows a group of people over time to track their health outcomes. Second, a case control study, which compares people who already have a specific condition to people who don't. A classic observational example would be tracking smoking habits and their link to lung cancer over a couple of decades. On the other side, we have interventional research. Here, you are hands-on. The researcher actively introduces an intervention, like a brand new drug, and measures its effects. The absolute gold standard for this is the randomized controlled trial, or RCT. This is where participants are randomly assigned to either get the treatment or a placebo.
Okay, the second major blueprint choice you have to make is between descriptive and analytical research. And the difference here is essentially what versus why. Descriptive research focuses strictly on documenting characteristics, behaviors, or trends. It tells you what is happening. You use surveys and case reports for this. For instance, documenting the prevalence of hypertension across different age groups. You aren't asking why they have it, you're just describing the facts. Analytical research, however, goes a level deeper. It investigates the actual relationships, causes, and effects. It wants to know why it's happening. The methods here involve cohort studies and heavy statistical modeling. A good example would be analyzing the underlying factors that are actively contributing to an increase in heart disease in a specific population. All right, let's do a quick midpoint knowledge check. Active recall time. This is a question straight out of the typical multiple choice test banks. What is the main goal of basic research? Take a second, lock in your answer. Is it A, B, C, or D?
Did you guess B? If so, spot on. It's B, to expand general knowledge and theories. Remember from our pipeline discussion earlier, basic research is all about expanding fundamental knowledge in the lab without worrying about immediate practical applications. If you nailed that, you're doing absolutely great.
Okay, let's pivot to Phase 5, research ethics. Look, methodology gives us the tools to discover, right? But ethics? Ethics provide the absolute fundamental principles to keep those discoveries safe. And no, this isn't just bureaucratic red tape. We need research ethics for some very real reasons. First and foremost, it protects human and animal subjects from harm, which is non-negotiable. It also ensures research integrity, honesty, and transparency. It actively prevents disastrous, unethical practices, stuff like data fabrication where people just make up numbers, plagiarism, or hiding conflicts of interest. Ultimately, strong ethics are what promote public trust in science. If the public doesn't trust the research is useless. And finally, of course, it ensures compliance with the law.
So, what are the actual pillars of research ethics? Examiners absolutely love to test these terms, so pay attention here. First is autonomy. This means ensuring participation is totally voluntary and strictly relies on informed consent. Participants must know exactly what they are signing up for. Second is beneficence. The study has to maximize the potential benefits while minimizing any risks to the participants. Third is non-maleficence. This is a fancy way of saying do no harm to your human or animal subjects. Fourth is justice. This ensures that the selection and treatment of all research participants is entirely fair and unbiased. And fifth is confidentiality. You have to fiercely protect the sensitive, private information of your research subjects. And finally, Phase 6, ethics committees. We know the rules, but who's actually enforcing them? You can't just start a study because you feel like it. You need formal approval before you even begin. There are two major gatekeepers you need to know about. First, the IAEC, or the Institutional Animal Ethics Committee. They oversee any research involving animals to guarantee humane treatment. They operate under strict guidelines like those from the CPCSA. Their entire job is to review proposals and ensure there is absolutely minimal pain and distress, and that using animals is actually justified in the first place. On the other side is the IHEC, the Institutional Human Ethics Committee, sometimes just called the IEC. They regulate research involving human participants. They make sure you are complying with good clinical practice, as well as rules from major bodies like the WHO or the FDA. They look at your clinical trials to protect participant rights, verify your informed consent process, and check your risk-benefit math. Now, when we're talking about the Animal Ethics Committee, there's one specific concept you absolutely must memorize for the exam, the 3 R's principle. These are the core operational guidelines for ethical animal use. First, replacement. This means you should use alternatives like computer models or cell cultures instead of animals whenever possible. Second, reduction. If you have to use animals, you use the absolute minimum number required to still get statistically valid data. And third, refinement. You must constantly refine and improve your procedures to minimize any potential suffering or pain. So remember that: replace, reduce, refine.
Okay, one final knowledge check to lock this all in. The 3 R's principle in animal research stands for what? A, B, C, or D? I literally just gave you the answer to this one, so I'm hoping you're basically shouting it at the screen right now.
And of course, the answer is A: reduce, refine, replace. This is the absolute cornerstone of ensuring minimal animal use and suffering throughout the scientific process. As we wrap up this explainer, I want to leave you with a thought to chew on as you head into your exams. We've talked a lot today about methodology, primary versus secondary, basic versus applied,